Participant.
A very good afternoon and welcome to the technical session of our webinar series. Today is our fourth technical session. Before we start, may I kindly request all the participants to kindly mute their audio. So once again, I welcome one and all to this webinar series of ours, that is Kindling Curiosity, Ushering Young Minds into Research, organized by Department of Zoology, Rajadhani College, in collaboration with KEYS, deemed to be university. And it's our fourth technical session today. May I now request Dr. Bandita Rath to present the address of our keynote speaker of the day.
I am honored to introduce the speaker of today's session, Dr. Nrisingha Dey, distinguished scientist at the Institute of Life Sciences, Bhubaneswar. Dr. Dey obtained his BSc, MSc, and his Ph.D. degree in biochemistry from the prestigious University of Calcutta, West Bengal. This was followed by his postdoctoral research work at the University of Kentucky, Lexington, and Iowa State University, Ames at the USA. He has been associated with Institute of Life Sciences since the year 2004, when he joined the organization as a lecturer, and where he currently holds the position of Scientist F. An eminent scientist in the expert in the field of biochemistry and molecular Huh? Am I audible now? Okay. He was also elected as fellow from West Bengal Academy of Science and Technology in the year 2017 and National Academy of Science, Allahabad in the year 2019. His current research activities are focused on molecular characterization of promoter for designing efficient native promoter and or chimeric promoters for enhanced gene expression in transgenic plants. Dr. Dey has over 60 publications in international journal of repute and has a number of patent and copyrighted material to his credit. His work has been cited in nearly 1,500 instances. He has also co-authored several books on the subject molecular biology and biotechnological application that are highly regarded and referenced in the field. Personally, apart from being a great scientist, Dr. Dey is an epitome, or rather Dey sir, is an epitome of humility and placidity. We are extremely fortunate to have Desa with us today to share his thought on research, a novel profession. I hand over the platform to Dr. Desa so we may hear from him. Thank you, sir. Thank you so much.
So please unmute yourself.
So it is very good afternoon to all of you. It is a pleasure for me to be a part of your activities, and today I'll be focusing activities, research activity as a novel profession. Uh, Research is a creative and sensitive systematic work undertaken to increase the stock of knowledge. It gives us in-depth and scientific understanding of how all living and non-living organisms exist on the earth, and so on. Now, why we need to do research? It is to perform or solve the problem that we don't have any solution, and to achieve the solution we have to perform or we have to conduct research to define the cause and effect and build evidence for or against a hypothesis. Now I come to my topic where I have to describe or I have to say something in favor of the research activity and to make the student population who are pursuing UG and PG courses in different colleges to opt research as their profession. So if we go back after qualifying the 12th standard exam, a pool of the 12th standard student qualified for the medical and other pool going to the engineering subject, and most of the students who didn't qualify medical or engineering are come under the basic sciences and doing some activities, research activities of other kind of activities. Now, I will promote or I will talk how a basic science student can be a more beneficial position in comparison to the medical or engineering student. Apparently, after qualifying medical, NEET, UG or PG, or engineering like JEE Advanced or JEE Main, those qualified students are really the cream of the society, and they have or they apparently very happy and very optimistic for their future. In contrast to this portion of the student who comes under the basic sciences, under the umbrella of basic sciences, are confused. Confused in the sense what to do after BSc or after MSc or after that profession or in that profession. So, if we carefully see the what is the real happening or real things happen after couple of years of qualifying in medical or engineering, or qualifying or coming under the basic science umbrella. See, if you observe, say after qualifying from even IIT Kharagpur, the IIT section like Kharagpur, Delhi, Madras, whatever, those IIT students are usually work under in field condition, stay apart from their family for a long time, and after under they usually takes a government level job. Most of them are taking the government level of job, and 80% of the above IIT qualified students work under private sector. Private sector means an electrical engineer takes their job in some electrical company like Philips, like cable company or whatever, and they always work under bosses, always work under bosses. So they are stressed, and their job are monotonous. Their job are monotonous. Similarly, the student who qualified MBBS, and then most of them only 5% or 10% going to the uh, PG degree, PG MBBS. But even after qualifying the PG degree in MD or whatever, this population of the student who come from the 12th standard population, they usually open as their profession, take the doctor or take to serve the society. They usually open their own chamber or works under state level hospital, and 2% of that population who become doctor gets really chance to work in national level hospital like AIMS or Jibban, and very few of them come to the international level. So if you see the fate of the student after qualifying medical NEET, UG or PG, or joint entrance main or advanced, after 10 years or 5 years of the downline, they their job are very stressful and monotonous, and they sometimes works under different bosses, and there is under different environment. In comparison to this sector, the student who comes from the basic sciences, who take the research as their uh, as their profession, they usually in better position. Better position means they can enjoy freedom. They can enjoy their own opinion they stick to their opinion, and have huge and have huge job satisfaction, compared to the doctor whose work are really monotonous after 10 years down the line, and also the engineer uh, who works in different field under different company and under different bosses. So if we look it in little deeper, you see in medical profession, the medical doctor and the patient are always 1:1 ratio. A doctor usually take care of a student, of a patient, single patient. If a patient admit in a hospital, it admits under one doctor. So 1:1, and if something happened to the patient, some casualty happened to the patient, doctor is the responsible person, and he sometimes beaten by the patient parties, or uh, is- apart from this, the doctor are worked under very less infrastructural facility, less medicine supply, less bed, less oxygen supply. So their work under 1:1 compare 1:1. If something happened to the patient, the doctor is get usually get punished by the peasant party. There is a chance, and also they work under very tough condition. And also the engineer also they work for just engineer and their job are in 1:1 ratio. Say a particular engineer is responsible, as we see in the figure, that that particular engineer responsible for running the machine. If something happened to the machine, something happened, so he is the responsible, and he get punishment or he gets something, something harassment from the boss, whatever. But in comparison to the researcher who works in laboratories or who works very independently and with a very good environment, as I mentioned before. So in research, it is scientists work in making transgenic plan. So in that cases, he can make lots of transgenic plant. If one plant die or 1,000 plant die, there will be no problem for that particular scientist. So say a part of the cloning. So uh, say in case of engineer, a person has to build a engineer has to build a bridge. So they will be given a number of the raw materials, number of these iron pieces, this number of bolt, this number of nuts, this number of welding machine, whatever. So a particular engineer gets on resources are very less compared to a biotic genetic engineer who need to get a clone. In that work he used tons of plasmid or many number of the plasmid, many number of the insert, many number of the enzyme molecule to get a one clone. So one to get one clone he can have billions or more than more uh, uh, resources. So in that case, researchers are better position. What I understand, I may be wrong, but in my conception or my perception, they are in better option. So coming to the next, where uh, say in this figure, say now I am considering the UG or PG student who comes under the umbrella of the basic student, how they can be benefited in long run if they take research as a noble profession. Say UG and PG student who come out from the university or sub colleges, they can take UGC or CSR or GET exam, get the net qualified student, and they did their PhD from any university, institute, or somewhere else. From that, they get the PhD degree. In some other part of the pool of the student who didn't get qualification of UGC or CSR, they can work as a project assistant in some laboratory, in some project, and they, after qualifying CSR, they come back again to the net qualified portion, and rest of the student who didn't qualify CSR or net, they can come as a non-net qualified student and they can have lots of opportunity. They can go and take the civil service. They can go for the bank jobs, business, enterprises, teaching. Lots of opportunities are there for the student who didn't qualify the net qualification. And you know, if a non-net qualified student gets civil service, qualifies for the civil service exam and becomes an IAS or IPS officer, under that officer, number of engineers and number of doctors usually work under the particular IPS officer. So there are lots of opportunities out there, and I'm giving this example just to promote or just to encourage the PG student or UG student under the basic sciences, just to don't get any frustration, keep keep thinking in positive way, and one day he or she will become the boss of the other subject. So Now come to the mainstream. After the net qualification, they usually get the PhD from the university, and then after PhD, they can go for the PDF abroad or at home institute. They can get the postdoctoral training or can accept the teaching or scientist job in India, or can accept a job in MNC or Indian companies, or start self company. So there are lots of opportunity opens after qualifying the PhD degree. Or there are lots of opportunities are there. And in addition to this, these students can be come under scientific network. So these students who come in this zone, they are all kind of friends, and they can interact with each other under a scientific network. And the end point of this, they can generate novel discoveries or challenging opportunities. So interaction, or there is an opportunity for this pool of the student can generate novel discoveries, uh, or challenging opportunities, and then ultimately that leads to the prestigious award, and also they can get they can nominate for the Nobel Prize, or most of them from this school gets the Nobel Prize in tons. Now you see these two, my colleague Dr. Sanjeev Sahu and Amresh Panda. They are working in our institute, and we are working together. And these two people by the Stanford University elected as a 2% top class scientist in the world. So this is a great recognition. This is a great recognition as a scientist. So the student, and they started their journey from the umbrella, from as a student under the umbrella of the basic science, and they opt as a research as a profession, and now they are getting a better recognition and prestigious recognition. So don't lose your hope or your faith in science or research. Come to the next. So there are several scope of the research. There are several scope of this research, and I don't go to each of them. And just as a main theme of this thing, there are hundreds of breakthroughs that have occurred and continues to occur in many fields of biology or biological sciences, and human civilization. For flourishing the human civilization, research is the only way. Now, so this is the different kind of researches in different aspect of the sciences, or different discipline. And this all of them are started from their journey under the from the umbrella of the basic student. So this is the discovery of the DNA structure, Illumina-based sequencing, human genome project. All the scientists involved who did their very good recognition in science in the world, they opt research as a profession, and they respect research, and ultimately they get good achievement. And all of them started their journey after finishing the high school from as a basic student. So now I will say something about the research which I am pursuing at ILS. So this is, the today is the due to the global um, global heat increase, global warming and water crisis. we need to make our food to be secure. It can be possible if we made some plant that have resistance under they have resistance for the heat and some water scarcity. For a plant to make such a modified plant, we need to express some of the gene or some of the information, external message or external gene uh, into the plant, and that can be happened through a expression vector, that is plant expression vector. And that plant expression vector which can be used to make modified plant or genetically engineered plant can be possible only through a good availability of the good expression vector. So my what I am pursuing my research at ILS. Just I want to share with you. It is not a good high level work, but from this work I have generated more than 12 Ph.D. and they are now settled in the USA. 9 of them are settled in the USA, and they have now become assistant professor or scientist in different university and colleges abroad. So just I'll show what are the type of work and how we are working at Institute of Life Sciences at Bhubaneswar. Now, so you know, for a gene to express in a plant cell, we need a promoter. We need a promoter. This red spot is a promoter. So this promoter drives the gene for its expression in the plant cell, in the cell. So we can compare this promoter as a railway engine which drives the compartment of the rail through a driven track. Similarly, this promoter, which is the engine of the gene, which is the regulator of gene expression, drives the coding portion of the protein into the plant cell. So my work is associated with the promoter. So like an engineer, they modulate or they modify the engine, making it either a very high speed engine which can drive much faster than a conventional engine, or to make this engine to drive under less fuel. This is the two objective or couple of objectives of the engineer, locomotive engineer, to make an engine to run faster on the railway track, or to make an engine to drive under less fuel. So my work is to just to modify this promoter, which is the engine of the plant gene expression, either to make it more functional, more stronger promoter, or more efficient, to increase its efficiency to express the gene, and also to make it work under different stress conditions. So you know, the rail on the track, its movement on the track controls by the different light signal. So when the signal is green, the train moves on the path. When the signal is red, the train has to stop in the red signal. Similarly, in the gene expression, there are lots of protein factor. There are lots of protein factor that is called transcription factor, which controls the activity of the promoter. Say, uh, say this is the promoter sequence. This is the promoter sequence, and this is the ATG, and this is ATG, and this is the coding sequence. And I told earlier that this promoter is a relay engine which drives the gene through the mRNA backbone, mRNA structure, or for its synthesis. Now, this promoter is situated on the 5' end of the gene, and if it is a chromosome, and a chromosome contains multiple number of genes, and each gene has a promoter. So uh, the right part of this chromosome is the eighth stop codon, and the left part, there is no end of the left codon. It comes to the very end of the chromosome. And each and every base pair in this portion is a long thread of the DNA are involved in regulating this promoter function that controls how the promoter will express this particular gene. So I repeat you that each and every nucleotide 5' of the promoter controls the function of the promoter. Now, say this is the promoter where the RNA polymerase bind, and there are several lots of promoter binding protein. There are lots of binding promoter protein binds to the vicinity of the promoter sequence, and this protein usually controls the polymerase activity, and that polymerase activity controls the promoter function. And in the left hand side, which I showed you in my earlier slide, that there are lots of DNA sequences, lots of cis factor, lots of cis element. Cis element is a small type of DNA sequence where the transcription factor are binds. So in that particular region, there are lots of cis element, and respective transcription factor bind to the cis element, and this transcription factor binding to the cis element, this cis trans interaction usually sends a message to the core promoter sequence, and that tells how the promoter will function. So there are lots of cis elements. So I put them in different color, and correspondingly, uh, this is yellow, this is red, this is blue, this is pink. So each are different, and each cis element has a particular transcription factor. So once this particular cis and transcription factor interact, they send a signal, electronic signal, and that controls the function of the promoter. So what we are doing in our lab is to just to change the cis element. We genetically modified the upstream part of the promoter. Do is replace the cis element. We modified the cis element and controls the binding of the transcription factor. And you know there are transcription factor, a group of transcription factor that works on the trace, like drought, some are heat, some are hormonal signaling. So in that way, we can manipulate, say by deletion of one, this is the native sequence, we deleted one of the yellow cis element. So this is the modified promoter sequence with a delta cis element, particular cis element. So in that way, we can modify the function of the promoter. So you know this is the mobile designation of the signaling pathway or promoter activity. So here is the highest signaling and the promoter activity we are obtained by changing or replacing the cis element from the 5' end of the promoter. Now, my lab is working with the pararetroviruses, and where we modified the promoter sequences from different pararetroviruses like cauliflower mosaic virus, fibro, peanut, whatever. 9 or 10 we are working, and each cases we isolate the promoter sequence, and then we manipulate the promoter sequence to make it to function in much higher efficiency or lower efficiency depending on the situation. Here I'll tell just a brief idea about the Petunia vein clearing mosaic virus. It is a colimoviruses. Colimoviruses, so it infects the Petunia plant and it makes some chlorotic disease and makes some disease of the plant, and it makes the plant to die. So this is a good model. Columophyrosis is a good model virus because it infects the plant and regulate the plant genetic structure. So I have taken this promoter sequence from this peanut chlorotic virus, which is look like this is the promoter sequence, and in the promoter sequence there are several cis element, mat box, then GRE box, then drop boxes, with key, whatever. So there are the cis element, which is the binding portion of the transcription factor. So we modified this cis element with the cis element AS1 cis element, DOP cis element, and make it a recombinant promoter or modified promoter which can work under different traces. Like when we increase the DOP cis element in this promoter sequence, and then DOP is a drought or abiotic trace transcription factor. So that modified promoter will work better under drought condition. So in this way, depending on the bioinformatics, we made different deletion construct of this promoter, and we get some of the activity or identify the which of the promoter fragment having the highest activity, but less lesser in nucleotide content. So our objective to get a promoter fragment having highest activity and smaller in size. So in this way, we check the activity of the promoter in different system. This is in transgenic system. This is in transgenic system. This is in protoplast. This is in agroinfiltration. And using the gas as a reporter gene, we make the gas activity. We measure the gas activity, and we found PV8, a promoter fragment of the peanut chlorostric full-length promoter, have highest activity compared to the CMB35S promoter, which is the most widely used promoter in the world. Now, we put that promoter on the GFP reporter gene, and we found that that expression of the GFP under that particular promoter is much higher compared to the other fragment. This is the histochemical staining of the PB8 promoter with gas gene, and we see the PV8 promoter has the highest expression. The intensity of the blue color determines the activity of the promoter. So PV8, we isolate the PV8 promoter having highest activity, and now we compare. This is the same thing. There we are getting the PV8 is the promoter fragment, and now we modified the PV8 promoter by altering the AS1 and W box cis element. This is the cis element position. We found that this cis element has direct involvement in that promoter activity. We did the footprinting, and we found that the protein, Wilkie protein and TGA protein binds to that promoter sequence that and control the activity of the promoter. Then once we confirm that these two transcription factor binds to that particular promoter sequence and controls its activity, and then we found how they control. Then we delete that. Now we made construct by deleting a particular cis element, AS1 element or W box element, or in one construct we mutate the only AS1, in other construct we made deletion of the W2 construct, and in some cases we made both construct and then we played in different in many many different ways, and we found that when we mutated, this is the original activity of the promoter. So when we mutated that AS1 of the green chair that I showed before, the transcription factor binding replaces the promoter activity. There is a loss of the promoter activity is if we mutate this particular cis element. Like if we mutate this particular cis element, the promoter activity also goes down. So where we made the all two deletion in the promoter fragment, the activity goes down. So initially this one is the native promoter activity. When we make it deleted, so these are the this activity of this promoter fragment goes down drastically. So in this way, when we mutated the three cis element, the promoter activity goes down. So in this way, we make it confirm that the AS1 and W box controls the activity of the PV8 promoter. This way, then we did the East2 hybrid assay, and we found the same thing. And this is the assay where we confirmed by overexpressing the TGA in the plant and within the plant. When we use these two transcription factor and overexpress it under in the PPS in P8 transgenic plant, when a plant expressing PV8 transcript promoter, when we expressed additionally or overexpressed TGA and OFG, we found the activity goes up. So these are the two main players where we can control the promoter activity, or we can express to get the activity of that PV8 promoter more in function. Then we have to overexpress these two protein. And if vice versa, if we want to shut down the promoter activity, we need to knock out these two cis element and these two promoter site. And similarly, these two are biotic stress responsible transcription factor. So this modified promoter can drive expression of a gene under drought or salinity condition. So this has been proved, and also we have used this promoter. We make a transgenic plant under the TSP, terrestrial secretory protein. It is a insecticide protein like BT cotton, BT. So we express this protein under that particular promoter, and we found that the epidopon insect, which is a leaf eater, their depth is very high. And see, this is the figure of the control plant, where the say in the control plant and in the transgenic plant, we place different larvae on this plant, and after 8 days of incubation, with plant and larvae of that insect, we found the control plant get highly damaged, and this transgenic plant, there is no damage. And this similar thing we have tested in rice, and this is the morphology of the insect larva after after the treatment, after the incubation. This is with the control plant. This is with the transgenic plant expressing TSP protein. Similarly, we express defensin protein under our modified promoter. We made transgenic.
So please unmute yourself. So please unmute yourself.
Sir, please unmute yourself,
Ah.
sir. Sir, please unmute yourself, sir.
Fine, sir.
Sir.
Fine. It's fine. Take it on.
So we express the human defensin protein, which is an antimicrobial protein. So we express the protein in plant, and then we get the extract and inject the salmonella infected mice. And we made the mice infected with salmonella typhoidium, and this is the control mice, and then we extract the protein from the transgenic plant and we infect it as a booster dose when and found that when we administered the transgenic protein, HBD protein, into the mice already infected with Salmonella typhoidum, that in case of the infected mice with the transgenic protein, their survivability is more than 210 hours. Or some of them are survived and they recovered from the Salmonella typhi trace, and they also come back to the normal life. But in cases where we put inject the Salmonella typhi in the mice and put the extract from the normal plant, the mice died after 72 hours. So in this way, we can control the expression of the foreign protein in plant for making either in transgenic translation product, or we can use it as a to control the gene expression in a metabolic pathway. We can identify a promoter, and then we can make it manipulate that promoter sequence by different strategy, and where we can exchange a portion of our promoter into the plant, into the plant, and replace that promoter sequence from our promoter by gene editing technology. So we are working on that right now. So this is a part of my research where I am working for developing novel promoter plant expression factor coupled to the novel promoter, and those expression factor are very efficient under different traces and also can be used in translational research. So what happened? So I think so. This is my group, and it is the lobby in front of the staircase of our lobby, and this is my group. So thank you for your patience, and any question I can take right now. So my question is that actually in my life also I qualified my HS exam. I got secured only 55%. At that time, my friends are they are either in medical or in engineering subject, or in some of the chemical engineering subject under Calcutta University. So I qualified my HS exam in 1983. Now, what happened after 1983 to 2020? So it is a gap of 37 years or whatever. Now I find those friends who went to the medical science or somewhere, they are what I said, that I am in much better in condition, or much better in position compared to my friend who qualified net GATE at that particular time. So my just want to pass this message is that whatever you get, either medical or engineering or basic science, don't uh, take it casually, take it very seriously, and respect your own subject, and you will be the winner in one day. So, in my talk, I just to give some idea of the scope, idea about the opportunities for the basic sciences. But in in medical sciences or in engineering subject also, unless the medical doctor or the engineer didn't take any research activity or didn't have any professionally love to the research in their own respect, in their own field, they become kind of they can make more money, but have less peace. So only research is the opportunity where a basic science student A medical student or engineering student can have their own self-satisfaction, and all are in science, and there is no end of the science. So if you work for science, if you work means if you love to research in science, there will be no end of the research, and there will be no end of your joy and happiness in that particular activity. I don't say that medical or engineering is they're really good subject, but unless you have of the research in that field also field too, your life become very monotonous and very hectic. So a doctor I know in my house, just in the locality of my, he comes at 8 o'clock every day and going back to 2 o'clock for lunch. Again come back 6 o'clock in the night, at the evening, and leaving at 10 o'clock. This is his routine, and in this routine he is prescribing. So, ma'am, my son I taking to the doctor when he was in 5 years old. He and kind of asthma he has same medication, paracetamol, some expectorant, some anti-allergic, and some something anti-allergic and some antihistamine. So after 12 years, I take him to the same doctor, same prescription. And also just recently I went to the same prescription. So I don't know how that person survived. He has money. He has money. But something apart from the money we need for our survival. Say a teacher, when a teacher teaches a student, a group of the teachers, say in my case, uh, if I go to my own school or colleges back in the Kolkata, my teachers just give me, they're very impressed because I am a creature of that teacher. So they're happy. So a teacher makes student. So I said that I make 12 students who are now in abroad. They are happy. So seeing them happy, I am happy. And this is something extra that we are gaining from the research. We cannot gain in the engineering field. I know one of my friend engineer, he has a big team and he has to roam around every night to the electrical, he's an electrical engineer, to ensure that electricity is intact in that particular circuit. So he have to visit whether it is rainy season or a winter or a whatever, he has to visit every tower at the night with his motorcycle, but because he cannot take his car. So he has to move around and have to check the locality of this thing. And sometimes under load shedding or under power cut, he is the main person to be contacted. So it's a very impossible time. Say in my case, I'm doing my research and my job at an institute, and I have luxuries here. So what I should do, make that. One second. Ah, okay. So, if I need, I can go around 12 o'clock to my home, get some lunch, pass some time with my family, with my son, and then I come back and to do my rest of the work. I can go up to 10 o'clock at the night. But I don't get those, my friend didn't get any kind of this relaxation. So I request all the students to respect what you are doing and give your 100% in that field, and you will be the winner. You will be recognized at one point of time in sometime later or sometime earlier. Student, engineer and medical student, they are getting their success in the early part of their life, and rest of the part of their life are little bit not they are not happy. But in our field, even there is no retirement of the scientists, so they can work for the society for their own. So in this way, we are So the students should take it very seriously, and we have to have respect, our regards to our profession, to the profession under the umbrella of research. So with this word, I want to stop here. So any question, any discussion? So this is, I made the slide this morning with my one of my postdoc student. Just I have some ideas, so I asked him we can put this way. So we made them slide. It may be something not particular or appropriate, but just I want to show the pathway the student, any student can take and can go for their highest regards in their life, most prestigious of prestigious award in their life. So we should respect, love, and trust our research. So any of the scientists who got Nobel Prize, you will see they come from the very basic sciences. Number of award are, I don't say medical and engineering or other subjects are bad, but they have to associate it with the research. Otherwise, a pure medical science or pure treatment of the patient, not give the satisfaction. They have to opt research. A teacher has to opt research. Otherwise, otherwise, life is not fulfilled. Will not be fulfilled. So, and this is my views. My student also agrees with these views. So that's I pass this thing.
Thank you, sir. Thank you so much for everything you shared with us today. And you just rightly pointed out that peace of mind is the most,
At
I can
one
say,
point of time, so madam, at one point of time we need money. We need money because we need money. But at one point of time, we don't need money. We need some happiness,
mental.
some pleasant, some shanti kind of thing, just to keep ourselves intact. But
Very true, sir. Very true.
and
Money won't solve all the problems.
and we can find this kind of happiness in kind of joy only if I get some good result in our research. So this way we can be involved and see how much.
Definitely, sir. Sir, can we take a few questions?
Yeah, please.
Yeah.
Please.
A student wants to know if he wants to prepare for NET examinations and he wants to pursue his career in research, how should he prepare himself from the undergraduate level?
Achha. This is a, I think this is the main, or what is the, this is the point, let qualification for this is the key. I should make a key point here. So if a student qualified NET exam, so he is set. He is somewhere, wherever he goes, is set. My teacher, in my, just apart from your question, I'll come to the point, to your question. My teacher, Professor R.K. Mandal from Bosch Institute, he told that I don't know what you will be doing after, once I go there and enroll as a PhD student. He told me that I'll give you some training, I'll give you some PhD degree, and I don't know you what you will be doing in future. Whether you will be a scientist, whether you will be whether you will be a bank officer or whether you will be a professor, I don't know. But I will tell you surely that you will ever sit ideally at home if you qualify PhD degree. So the getting the PhD degree, the main question is to qualify the NET exam. And now, NET exam is the key for everybody's success. If a person did very high percentile in his MSc or MPhil, MSc classes, unless he qualified the NET exam, there will be no, there will be chances are less. Chances are less. For qualifying the NET exam, my suggestion is to, there are questions available, right? Questions available for last 15 years question, solve that questions. There are some guide are available, guidebook are available, and also some coaching classes are available. If that coaching classes just they teach something, but they make test. Lot of test are there. So that gives their mentality to be oriented in that particular exam. So test, and you cannot give the test in your home. In your home, say in case of Akash Institute, what they are teaching they don't know, but they are going to test every weekend, and there are multiple test national based course test. So once you bring that affection or that interest for taking the test, not for getting the score, but taking getting the sense how the exam passes. So you have to answer 720 whatever questions in 1 minute per questions. So you have to adjust your timing for that answer, and in that way there will be a sense will develop which question to attempt, which question is not attempt, because there will be a negative marking. Probably there is a negative marking. So a particular basic or smart student only answered the right questions, picked up the right questions and answered it. Don't take the haphazard questions or random questions. So in that way they can succeed, and they also discuss who already with who already qualified the NET exam. So NET exam is the must exam. NET in our time, in my case, the CSR exam was the first time exam. So first time I didn't know about the CSR exam. So I didn't give when I was in MSc second year. So after qualifying MSc, I worked as a project assistant in Rajadhani Science College in a chemistry department. Then I take the exam in the second year. The worst exam I given in my life. Take it granted. The worst exam. I didn't prepare because I was working as a project assistant. So I take the exam and I didn't know how to answer, how to. Then that is the worst exam. Without preparation, without it. But the marks come, I qualified under CSR category. Why? Because it gives a percentile code. Percentage of students who secure less mark compared to my mark. So this is the ratio they are calculating. So I know most of the students who just have a idea that I don't take the exam this year, I'll take the exam next year. But to them, my suggestion is that take my point, take my experience, you go and get the test. Whether good or bad, it doesn't matter. It matters that how many students gets poor marks or lesser mark than your mark, and that will give you a percentile. And if you cross the percentile of a particular cutoff, you will be selected. So, for suggestion, the student should give test like corona test to identify whatever as many as possible test take. Then talk 15-year question, and also the talk to the different student who already qualified. They have some experience, and also take the exam. Don't excuse to bypass the exam, and I think it will happen. And once it any student come and CSR qualification already, there are lots of opportunities are there. And any institute, ours like us, Institute of Life Science, they're taking around 40 students per year CSR qualification. And there are so many institutes in the world, like Institute of Life Sciences. So lots of opportunities are there.
Thank you, sir. Hope the participant have got his answer. Bandita madam would like to ask something.
So usually PI brings a project, sir.
Ah,
They bring a project and they take up a PhD student.
ah,
There are some students who are like specifically into biotechnology, who are specifically into microbiology. So they would like to know whether they can have a project and they can, like they themselves, they can then go to a PI for that, for having their PhD, sir, without going through the net, sir. If they can avail themselves the project by themselves.
There are a couple of government offices like BIRAC or something, but somehow he has to attach to someone.
Yes.
Actually, the concept of PI takes student with a vision that he or he takes students or he or she takes student under a project, and in that project he has lots of experience, 10-20 years experience in that project, and he knows what is the point to be addressed, and once it addressed, it can be addressed in a minimum period of time, 2 years, 3 years, and that can be publishable, and a PhD can be given or get by a student. So in that orientation, always a PI tech student. But I think no PI in the world tech student haphazardly or just as a random. No project, no idea, tech student. So a student when joins to a PI group, PI has a 10-20 years experience of that project. He knows which particular pocket he has to work to get a publication or to get a better work and get a PhD to the student. But in the other hand, the students sometimes don't like that. They told that I have much better idea than this. Everybody has a better idea, but PI has the best idea for that project only. He knows the pocket. If you say my home is somewhere in Nalkochok, so I walk down every time to Nalkochok and this. So I know each lane and bylane of that road. So I know which way if I go, I can reach that point much easier. But the students say, why I go this way, I go that way. So sometimes makes it is sometimes happen, sometimes not happen. So better to just have a trust, faith on the PI. And all PI works for the student, not for himself, for the student only. Say many PI has already 20 PhDs in their career. So they don't know PhD. But only for the sake of the science, they are taking the help of the student and they are giving their progressing their science. So, but many cases there will be a fight between student and PI. Sometimes PI said, or student said, I'll resign from your lab, I'll go somewhere. So in that way, it is my viewpoint. From my viewpoint, it is the loss of the student only. So better to stick to the PI. PI has definitely some idea, and PI has the responsibility to take the student to take out of the river, to swim out through the river. But and without net or gate, there are a DBT project, BIRAC project, there is an option. If you have some brilliant idea, you can come, take some project. But the success rate in that project is very minimum. So, and it is very challenging to get it. If a student have an idea, that idea has to be approved by a senior level scientist, a group of panel of senior level scientists in the task force. Then it will be approved. So it is very hard, very hard.
Thank you,
But
sir.
Thank you.
Thank you so much, sir.
Thank you, sir. Definitely, sir, experience matters, and that is why we are really fortunate to have you today to share
no,
all your
I'm always
experiences with
available,
us.
always available. When you ask me, I'll come to your and college or
Thank
university,
you. Thank you.
whatever. We are free. ILS is free to do this kind of work, and it is our pleasure.
Thank you, sir. May I
Okay,
now
thank you.
request Dr. Rashmi Mahapatra, madam, to kindly
I know,
propose
madam. Hmm.
the vote of thanks.
Okay, thank you, madam. Hmm.
Certainly, it is a great pleasure to propose vote of thanks for such a wonderful session. At the outset, I would like to extend my heartfelt thanks on behalf of the entire organizing team to our keynote speaker of today's webinar, Dr. Nrisingha Dey, for his excellent deliberation on the topic entitled Research, a Novel Profession. Sir, your session encourage the students by telling the importance of research, how the research is a really noble profession. And one should not go for engineering, medical, or do not get upset for not getting a seat in medical college or good engineering college. Very well, we can go for research. We can do a lot of developmental work for the entire country, for the globe, for human being, for the society, to get the sustainability of the environment, for food, for agriculture, for any health related issue, anything we can do. Highlighted different scopes of research activities by citing different examples. You have also highlighted about the advancement of technologies and tools and techniques so that we can do better and better research. Thank you so much, sir. So you have shared in your insight to with our audience. They are young mind college students regarding the field of research. I hope they will definitely get encouraged after your talk. After listening your talk, definitely they will contact you. We got the opportunity to listen from you, though I talked many times, not research related things. I have never seen this side of you. I have only seen that you are very hardworking, very humble person, very soft-spoken person. In that way, I have great regards for you, sir. And today, really, I am feeling fortunate and grateful that I got the opportunity to listen from you. Thank you so much, sir. Then here, one thing I'll add, sir, I'd like to offer my thanks to administratives and faculties of both the institute, KISS as well as Rajadhani College. And I'm really thankful to Madam Sasmita Sarangi, Madam, only because of her and because of her interest to collaborate with us, we got this platform, we got this opportunity to listen from eminent and another thing is, here I would like to thank my friend Swapna. She had joined from SRM University in this platform. I had requested Swapna. She is my best friend when I was in SRM University, Chennai. Throughout the talk, she's present, and really, I'm very thankful to Swapna. She has she has kept my request and joined in this platform. I'm also thankful and grateful to Nanocha sir. He has joined from IBSD. I saw he joined, and maybe because of some work he left. Again, he joined. Thank you, Nanocha sir, for listening this talk and for request for keeping my request.
Last but not least, a big thank to all our participants, especially students throughout Odisha, for participating and fulfilling the purpose of this webinar series. Thank you, dear students. Thank you, one and all. Thank you, sir. Namaste,
Thank you, sir.
Thank you,
Thank you,
all.
sir.
Thank you. Thank you. Thank you.
Thank you.
Thank you. Thank you. See you later. Thank you.